Today, Steve Hanson is going to give our second. And I do want to say these are the first Wednesday every month, unless there's a conflicting event. So next month, I encourage you all to come to the Fabian Garcia Fort Farm Field Day. It's going to be the first Wednesday next month. So that's going to take the place of the Chile Chat. And you can go and hear all about the Chile Pepper Institute, the teaching garden, and other research projects are going on at the station. However, the month after, July, Dr. Philip Lujan is going to be talking to us about chili pepper diseases and disorders. A very important talk, very great information. So I encourage you to join us the first Wednesday in July. But for today, is everything set, guys? Yeah. Oh, we're all awesome. So I'd like to introduce Dr. Steve Hanson. I've had the pleasure of working with Dr. Hanson for many, many years. He's a professor in the entomology, plant pathology, and weed sciences department. He's going to talk about some research that he's done in biological controls. And Steve, I'm going to let you introduce yourself further. Thanks.
All right. Well, thank you very much. It's a pleasure to be here. And before I get going, just two things to keep a check on myself as a professor. Number one is the time. We've got till this ends at 11:30. All right, so an hour and a half, I promise I won't go over time as professors are sometimes want to do. In fact, I don't have anything near an hour and a half of material to go over. So what I'm actually hoping is that everybody present in the room and those online, feel free to speak up, ask a question in the middle, interrupt me at any time. And if we go off on 5 tangents and I only get halfway through my slides, we may actually have a better chat than if everybody just sits quiet and lets me ramble. The group discussion is usually 15 times better than just sitting back and listening to me. And then the other really quick thing, Denise, are we connected with people online? Can they hear us? All right, fantastic.
We can hear you, Steve. This is Ed. I finally got on. I can hear you.
Perfect. Glad that you're here, Ed. All right. So as Denise mentioned, I'm in the entomology, plant pathology, and weed science department here. We're a little famous on campus because our name is, we've got the longest name of anybody on campus. I've been here just a little over 20 years. And all of my research has been focused on reducing losses to pathogens. So when we think about profitability of a farm, the basic equation is you take what the proceeds you get from selling your crop minus the inputs you provide or you put into growing your crop. And then that ends up telling you whether you're making it or not. And hopefully you're making it well. I know the last 20 years since I've been here has always been a challenge for chili growers. The reason that I'm so interested in disease is that individual fields vary. Some fields have no disease, some fields have a lot of disease. But and Stephanie can help and Soon can correct me if I'm wrong, but I think on average, chili growers probably lose about 20% of the crop in a given year to disease. So my focus on reducing disease losses is if we can fix plant disease, that's sort of an instant 10 to 20% boost in productivity and potentially with no extra inputs. So I see that as a way to improve the livelihood of chili growers and the quality of the chili that you guys are able to deliver to the market. And this, I think, is kind of in line with all the other major goals that the chili growers have. Reducing input costs, hand labor is a big one. We would love to get the same yield from a chili field with reduced labor inputs, which is why people like Stephanie have been focusing on mechanical harvest. But I think the big point is that my research program is really oriented in the same direction, trying to improve productivity for chili growers by increasing yields without No, you're good. That's perfect. Without increasing input costs. So I'm gonna share out some material that I presented at the New Mexico Chile Association meeting this past February. And right off the bat, to make sure that I don't forget at the end, I've got to give credit to a lot of the people that work in my lab, and there's pictures and names at the bottom. Erica and Jessica and Maria were all undergraduate students that helped with the research this year. So they got, as undergraduate students working in the lab, they not only took care of washing all of our dishes and watering all our plants, they learned how to make media, and each one of these as an undergrad actually did a little independent research project, which was really neat to have them do. Max Radianenko and Serjana, both very experienced people who and very talented, and they have been with me for quite a while, and they've also helped a lot with the research I'm going to present. I would love to say I do a lot of this, but really my main job is directing them and letting them get lots of cool stuff done in the lab. And so I make sure that I get them credit up front. Then, all right, so today, my group actually, we work on more than one disease, but today I'm going to focus on Phytophthora. And since Phytophthora is a soil-borne disease, even though I'm not going to talk about verticillium or fusarium or rhizoctonia, most of what I'm going to talk about will apply to these other soil-borne diseases. But up here, I've got a couple of pictures of Phytophthora chili wilt in farm field. One kind of close-up shot on the ground on the left. And then there's a high school student, Devin Chesley, that actually flew a drone and took some pictures for me. And I think he's going to be in here at NMSU in the fall, and maybe we'll be able to get him into my lab. So these are kind of classical, typical symptoms for Phytophthora, and all the growers in the audience actually recognize this better than I do. Every time I'm fortunate enough to walk around with a grower or a talented field person like Ben said, they actually teach me more about the symptoms. But when we get obvious slides like these, it's really interesting to, or really obvious to see the symptoms. Now that actually, no, we're all good. The caption at the bottom here, the whole Chile Association meeting this year started with a section or a session on Phytophthora. And the very first talk in the very first section or session of the Chile Association meeting this year pointed out that we've been dealing with Phytophthora for 100 years. It was first described in 1922, I believe, and soon can correct me if I'm wrong on that. But basically, we've been living with Phytophthora causelosas for 100 years without very much relief. Here and there during that 100 years, we've had brief periods where we were able to control Phytophthora a little better. And this was with some of the synthetic fungicides like Ridamil. Unfortunately, that relief didn't last very long because Phytophthora has evolved to overcome every fungicide that's been used to treat it. And so things like Ridamil and Quadris today don't provide very much relief. So we're still 100 years on trying to figure out and fix this problem. All right, I'm gonna jump to the end, and these are kind of plot spoilers. But even before I show you some of the data and some of the experiments, I want to put fresh in your mind the conclusions that I'm going to come back to at the end. And everything I'm going to talk about today are sustainable approaches to disease control, which are mostly organic biocontrols or other organic approved measures. And so sustainability is a big word today. And I think it's an important point that we want to focus on going into the future. Not only do we want to improve yields, but we want to make sure we're doing it in a sustainable way. So with every sustainable method, everybody's going to be familiar with things like integrated pest management. The integrated approaches are key. So when I get into some of the research, I'll talk about how some of the different biocontrol agents perform, and I'll show you slides that have where you can see symptoms reduced by this agent or that agent. But I want to put the idea in your mind that as we're looking at these, we test them individually one at a time in the lab. But we are very unlikely to find a single biocontrol agent that's going to be a silver bullet and fix all of our problems. And the lab research where we check these one at a time, our ultimate goal is we screen one at a time to find the individual agents that have activity. But our ultimate goal is to produce combinations of agents that will act cooperatively or synergistically so that the total activity is greater than any one of the individuals. And this is very, very similar to everything else that happens in a farming operation. With herbicides or pesticides, you have to rotate modes of action to avoid resistance. And then sometimes I give the silly analogy of fertilizers, where it's not nitrogen, it's not phosphorus, it's not potassium. None of those three individually are a silver bullet. But when you combine all three into NP and K and deliver all three at once, that is where you get effective fertilizer action. And so everything I'm going to talk about today, even though we're going to look at individual agents, we really want to figure out how to combine them at the end. The next point is thinking about sustainable methods or genetic resistance. And science researchers, we've got breeders working on genetic resistance, people like me working on biocontrols and other sustainable methods. And sometimes we get divided into camps, and would you rather have genetic resistance or sustainable methods? But in the spirit of the integrated approaches, I'm going to say, why not both? And if we get sustainable methods working well, then the genetic resistance will just work even better. And if we have genetic resistance, then the plants get a little less sick, there's a little less disease pressure, and that's going to help the sustainable methods work better. So again, with the integrated approach, these are not going to be a one or the other, but they're going to go hand in hand and actually complement each other. Right, so a little bit of background on the biological control. This is really making sure that we have the healthiest, happiest plant we can in the healthiest, happiest environment that it can live in. And this is really analogous to a lot of what we hear in the news today about human microbiomes. Everybody on the news likes to talk about your gut microbiome. If you've got beneficial bacteria in your gut, you're happy. If you get sick and have to take an antibiotic, that might clean your gut out and you might have an unbalanced biome in your gut. And that will make you unhealthy and unhappy. But if you do something like eat yogurt or take a probiotic, it's a way to restore balance to your gut, which hopefully returns your health and your happiness. So the plant system is exactly the same. And the soil that the plants grow in is inhabited by billions of different microbes, bacteria, fungi, nematodes, and all of those. And I think it's too small to see on this slide. I'm sure the people in the room can't read it. But if you look at some of the arrows, we have the microbes interacting with the plant interacting with the microbe. That interaction goes both directions. The beneficial microbes interact with the pathogens directly, and indirectly, the indirect interaction is the microbes interacting with the plant and making it more or less resistant to the pathogen and all of that. And then the next factor is all of this is happening in the context of the soil, which is where all the nutrients are, and then the bigger environment, which would be the temperature and the irrigation. Is the soil wet or dry? So it ends up being a very, very complex system. And for better or worse, the complexity there gives my lab a lot of stuff to work on. But it also presents a challenge. We need to find an efficient path that we can get through some of these complex questions to try and find some of the key actions where we can influence things like pathogen levels and pathogen activity. So I've taken the liberty of going from this really complex cartoon that's too complicated to read, and I've simplified them down. So in this really simplified cartoon, we'll call this sort of a natural state, where we've got a happy plant living in a natural environment that contains both beneficial microbes and pathogens. And in this case, the beneficial microbes and the pathogens are about equal levels, or we'll call this natural levels. And in this situation, we can actually have a healthy plant, and we still have all of these interactions. The beneficial microbes are interacting with the plant, and the plant is interacting with the beneficials. The plant and the pathogens are both interacting with each other in both directions, and then the beneficials and the pathogens are interacting directly with each other without the plant in the middle. Now, when the system gets out of balance, we have the pathogens coming to dominate the system. And so the pathogens grow and the beneficials shrink, and that leads to an unhealthy plant with a lot of disease. Now, unfortunately, modern agriculture actually encourages this sort of an imbalance. And the reason that agriculture encourages this is we're growing plants in a very non-natural system. In particular, we're growing in high-density monoculture rather than a field of diverse plants. Since we need to grow chili by the hundreds of acres, we don't really have a choice. We can't plant 17 different plants in the field to get a nice, diverse, stable ecosystem. So what we have to do is try to find a way to avoid unbalancing the microbiome and encouraging the pathogens, even though we're growing plants in sort of an unnatural system. So our potential remedies to fix this are things that we've done in the past, like using pesticides. And if particular fields get too bad, pathogen levels get too high, sometimes we just abandon the fields and try to find some new ground that hasn't had chili in it, and hopefully that will be disease-free and we can go on for a little while. Or one of the other option on here is the one that my lab is focusing on now, is seeing if we can sort of rehab the field, and in particular restore balance to all the microbes in the soil, which means pushing the pathogens back down, encouraging the beneficials to come up. And if we can restore that balance to the microbiome in the soil, then hopefully we get the beneficials up, the pathogens down, and we can return the situation to growing happy, healthy plants. All right, and I guess I'm kind of through the introduction, and since we have a lot of extra time today, this would be a good time to maybe ask if there's a question.
I have a couple questions. Sure. Our attendees on Zoom. Way back at the beginning, now it's a little sorry. Hello, Dr. Hanson. I'm in zone 5B to 6A, Wisconsin. I want something that eats fungus gnats. I white flies, they ruin my chilies. Any opinion on mantids?
Mantids. Right, one of my talents is admitting when I don't know something. And control of insects is outside of my expertise. The pathogens, the fungi, the little bit that I do know is I think the mantids and other predators are a great idea. And the gnats in particular, we don't really have as much of a problem with them here in the desert. So my experience is going to be even less and very, very low on that end. But I think I'm hearing Stephanie say that we will get an entomologist on the Chile Chat soon.
Yeah.
In the what I can say is in the world of biologicals, I think my email address should be or could be put in the chat. Some of the companies that I work with that develop the biological controls, I know that they're looking at biological controls for insects. And I would be happy to field an email from you and pass it along to some of the experts in the field. But that question is a little outside of my expertise.
I have another question
All right.
from our attendees online. This is from Matt, saying that 20% of production is lost to disease means that removing disease loss would return that 20% production. Assumes that loss due to other factors would have been lost to disease. Loss due to other factors would not have been lost to disease. Does it not? Is there evidence to support that?
Yes, and your point is well taken that I've presented a really simplified scenario. Different fields are different farms are different, and when and I'm speaking in simple averages. But on average, we really do lose about 20% of production to disease in a given year. And as for evidence of when this has happened in the past, when we get a breakthrough, like we discover a resistance gene and we release a new resistant variety that we didn't have the year before, or we discover a new pesticide that controls a disease that we didn't have the year before, we really do see those disease losses disappear and productivity does go up when that happens. So yes, these are simplified kind of least principle models, but in reality, these things really do work. When we have a big advance like discovering a resistance gene, releasing a resistant variety, or releasing a pesticide that alleviates a problem, we really do see the boost in yield. And then we actually get some of the funny knock-on effects. Does that saturate the market with overproduction? Do people get encouraged to produce less acres if they can make the chili more efficiently so that they don't saturate the market? So there are other complications to this, but on the first principles, if we could reduce disease losses, we really will increase productivity and yield.
Can I jump in?
Sure, please.
So that was my question, and I'm just having a hard time wrapping my brain around. So I 100% understand what you're saying, that if you reduce disease loss to disease A, that will increase our yield. That makes perfect sense. The question is more, if we reduce disease loss due to disease A, are we going to recover all of the loss that was due to disease A, or is there reason to believe that because this particular plant, the individual plants in the field, were weak to disease A, and that's why they were the 20% that were lost, are they also going to be particularly susceptible to disease B or to pest C, and therefore you're not going to really recover all that 20%. Because they're the quote weak plants, they're going to get lost to some of them are going to get lost to something else as well, and you're only going to recover 15% of your 20% or something like that.
Yeah, sure. So that's a really excellent point. And this is, I'm gonna come back to these systems all being integrated. In the simplified models, like the ones I'm starting off showing, we all like a yes, no answer, and we like linear things where Phytophthora causes this much loss. But things are rarely linear, there's rarely a single connection. And what we're really dealing with is an integrated system where we've got a web. So if we reduce Phytophthora losses by 20%, your point is well taken that we'll increase the number of healthy plants by 20%. And that leaves a bigger population that could be infected by other diseases and things like that. So I won't guarantee that if we measure Phytophthora really carefully, and we decide that it's reducing yields by 20.7%, and we fix Phytophthora, I won't guarantee that yields would go up by 20.7. We may get Phytophthora out of 20.7, but when we increase the number of healthy plants, we essentially set a bigger table for the other diseases and the other pests. So in principle, it's really difficult to put a number on that. Getting rid of Phytophthora, or even going back to evidence that this works, evidence that we have from the past, when you introduce a resistance gene, a resistant variety that escapes a disease that hammered you the year before, you absolutely see a yield increase. Whether you get 100% of the potential increase or whether you lose part of that because you set essentially a bigger table, provide more healthy plants that could be potential targets for other disease. I think you're probably right. So, and again, I can't put a number on it, but I'm going to say that if we reduce Phytophthora losses from 20% to 10%, maybe we raise yields by something like 6%. I think the general principle that we're going to step forward is unquestionable. That what's deciding the amount and whether or not we can put decimal points on the size of the step forward we take is probably not possible. Does that help?
Yep, that's exactly what I wanted to know. Thank you.
All right. Well, that's an excellent point, and I love talking the details like that. See, we have Ed Curry there.
Yeah, Steve, if I can, I just was going to throw in two things. Number one, doing this is my 54th crop here in the States. And if I count Mexico, it's up 11 more crops, and we're doing that in Sonora now. Our experience, number one, I'm going to go back to the question on, does it affect insect pressure if we're biologically sound? And I can emphatically tell you, yes, that my experience has been the more healthy the soil, the more healthy the biology, that your insect pressure is just naturally less. And Dr. Walker knows this very well. And her several have worked on this through the years, and even down to all the sugar beet leafhopper and the issue with curly top. The one thing we do know is if we got good, healthy soil biology, is it 100% answer to that? No. But is it a marked increase of improvement? Yes. Hard to put a number on that. But I want to make clear that John Kemp with Advancing Eco Ag has clearly stated that in his work, they've seen a lot of improvement, especially in the Midwest and the far Northeast in vegetable crops. They were actually able to almost do away with certain insect pressures just by soil biology and health. I myself, I want to throw this in, Steve. and you know this from working with me, but we've actually had better luck controlling Phytophthora in an organic farming system than we ever did with trying to control it with all kinds of chemicals, and you listed some of them, some Ridamel Gold, some of the common ones that we know. We've actually, over the years, the last 12 years, under a biological warfare plan and farming organic, when we got rid of herbicides and pesticides, we didn't have as much phytophthora trouble. And I believe it, my personal thought is it has something to do with those were hurting they were hurting what was going on in the soil. We were killing some biology with herbicides. And if you think about it, if you got something out there strong enough to keep a weed from coming up, you probably got something out there strong enough to hurt some biology. Now, I got growers that are farming totally the original way with lots of pesticides, lots of herbicides, and making tremendous yields. But on my farm, where I've been in Chile many, 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 many years, we have found that to go organic and back off, yeah, we don't yield quite as high, but we're also just in strict seed production. And so it's kind of a little different. But we also see that we just have a marked improvement and less incident of Phytophthora, not 100%. So I just wanted to throw those in. And in your quest about how much does Phytophthora affect yield, and we got to the point that we knew we were going to lose a certain percentage of the crop. 2011, 2010, respectively, those 2 years, I had 300 acres of chili. I lost half of it 2 years in a row because monsoons were heavy. Phytophthora took half of it, not hail, but Phytophthora. And literally, if you've added those 2 years, even with the expenses less in those days, that was well over $1 million hit. And that we were just common with it. And it was about that time I started switching back to organic and figuring out that with our chicken compost and those enzymes, that we seemed like we had a little better chance at the warfare. So I know I can
All right,
do
well,
or both.
thank you for those points, Ed. I really happy to hear those. And I think when I was answering Matt's question, I was trying to say that we may not preserve all of the Phytophthora yield because more healthy plants might be present for other pathogens to take advantage of. But you bring a good point up, Ed, is that if we improve the overall health of the plants, we may actually sort of indirectly make them more resistant to other pathogens as well. And the things I'm thinking of specifically are we commonly see plants that are simultaneously infected with 2 pathogens. And if we take one of those, if we take Phytophthora out of the mix, then the likelihood that we get extra damage from a co-infection with Phytophthora helping Fusarium or Rhizoctonia goes down. So these are the complicated interconnected web that make it difficult to predict exactly how much of the yield we'll retain, and will retain all of the yield that we get by suppressing Phytophthora. We may have more plants that are potentially attacked by other pathogens, or on the upside, we may actually have more healthy plants that when we remove the stress of Phytophthora, we may actually reduce exposure to some of the other pests and pathogens as well. So what I will say, it's a complicated system, and it's really, really hard to put a number with a decimal point on the answer. But the general theme of keeping the plants happier, healthier, a beneficial biome in the soil, plants will overall just simply do better in that situation. And then suppressing individual diseases within that certainly leads to good jumps in yield. So other questions?
Yeah, there's a question from Juan. What about soil metagenome balance or holobiome?
Okay, the metagenome and the holobiome, these are kind of technical terms. And they are things that my lab is actually doing right now. And I'm sorry, the name was
Juan.
Juan. I hope that you and I are, I hope that I'm using the terms the same way you are. The soil metagenome is something that we measure with a genome scale DNA sequencing machine. The way we do it is we literally extract all the DNA in the soil. We run it through the same kind of machine that 23andMe uses to sequence a human genome. And by doing that, we can look at the sequences and we can predict which bacteria are in the soil. So that's a metagenome analysis. The hologenome analysis is, it's kind of semantics, but it's sort of the same thing. But these are tools that we actually use to look at the microbiome in the soil. The reason these tools are so important is that traditionally we would look at the microbiome by culturing it or plating it out in petri dishes and trying to identify individual members of the community that way. And what people have known for probably close to 100 years is we can only culture a very small proportion of the organisms in the soil. So if we try to look at, if we try to characterize the soil microbiome by trying to pull individual members out, grow them and examine them, we probably only look at 2 to 5% of the biome. But when we extract all the DNA that's in the soil and do a metagenome analysis using deep sequencing data, we can actually identify the organisms that we can grow as well as the ones that we can't. So when we look at soil microbiomes now, our preferred method is to do this deep sequencing and look at the soil metagenome or the holobiome or something like that, because that allows us to see everything that's there and instead of being limited to the things that we can culture. This technology, to get more directly to your question, this technology is kind of in its early days, and as you can imagine, we get lots and lots of data, and it's really hard to figure out how to interpret it and get things out of it. But some of the things that we're able to do are kind of relationship analysis, that if we take a healthy field and compare it to an unhealthy field, then we can look for members of the microbial population, members of the microbiome, see who's up in an unhealthy field, see who's up in a healthy field. And this lets us start identifying, characterizing who the beneficials are, who the negative microbes and the negative members of the biome are, and things like that. So this is a really active area of research, and microbiome analysis is big today, and it's only going to get bigger in the future.
One from Chris. Professor Hanson, do drought conditions affect the prominence of the primary pathogen you mentioned earlier?
Yes, Chris. The environment is part of this interconnected web, and the environment affects every disease. And even if we don't have any disease, the environment alone has a tremendous effect just on the plants. Same field, same plant variety, 2 different years. 1 year is wetter, 1 year is drier, 1 year is hotter, 1 year is colder, same amount of moisture, but it comes earlier or later. All of this has a profound effect. And in particular, I'm going to flip back to the first page with the Phytophthora symptoms. Phytophthora is a disease that's heavily influenced by water. Some quirky biology trivia. Everybody likes to think of Phytophthora as a fungus, but now that we're in these days of sequence-based characterization and metagenomes and all that, People have actually figured out that Phytophthora is not actually a fungus. It's actually more closely related to things like algae. So we call it noomycete. It's more closely related to algae than it is to fungi. If you recover it from a plant, grow it on a petri dish, it looks like a fungus. That's why historically we've lumped it in. But its relationship to the algae, I think, has something to do with its water influencing the disease. Phytophthora needs heavy water to survive and propagate itself. And in fact, the infectious unit in the field is a little spore called a zoospore, which actually has flagella that let it swim in water. So in a dry year, we will get 1% or less of the plants infected, and it'll just be bad luck for a plant that lands on a spot that has some active Phytophthora in it. But when we get a lot of water, particularly a monsoon that leaves standing water in the field or water flowing down the roads, then Phytophthora flourishes. It reproduces really well, survives really well, grows really well in wet soil, and the spores not only get carried by the fluent water that flows down the rows, but those zoospores can actually swim around on their own too. So this is a disease that just is tremendously exacerbated by extra water, particularly late in the year. So chili producers down here in the desert Southwest, we can have a really, really good looking field at the end of July. We can only have 1 or 2 of the plants infected with Phytophthora. But if we get 2 monsoons that leave standing water in the field for a week, we can go from those 1 or 2 plants to losing half the field, because the Phytophthora being water aided will spread that quickly and cause that big a problem. Not all diseases are as dependent on water as Phytophthora, but it really is the environment, the temperature, the water, everything affects all diseases, but Phytophthora in particular is especially influenced by water.
Ed would like you to mention the study that me and Dr. Thomas relationship between Phytophthora in 2008.
All right, so here is another interconnected web. We'll come back to the Phytophthora picture here. The study that Ed is asking about is one that was done by nematologists in our department, Steve Thomas and Jackie Trojan. And they looked at the prevalence of, or the severity of Phytophthora disease when nematodes were present or absent. So I've been giving a lot of really long-winded answers, so I'll keep this one short and simple. The nematodes present caused more severe, or was associated with more severe Phytophthora disease. And the simple interpretation is that the nematodes feed on the chili roots, and the particular nematode they're talking about here is the root knot nematode. They actually penetrate the chili root, and the nematode lives inside of the root. So what happens is every time a nematode penetrates a root, that leaves a wound and probably makes the plant easier for the Phytophthora to infect. So here's another example of our integrated web where we can worry about Phytophthora on its own, but it's definitely exacerbated by having nematodes in the soil. So great points. Thank you.
There's a question from Cecilia. Is there a reduction in fields using drip irrigation as opposed to those fields utilizing flood irrigation, or reduction of Phytophthora?
Reduction in Phytophthora if you switch to drip? All right, and the answer is yes. Phytophthora being such a water influenced disease. The better you could control water, the more you can reduce disease. So drip irrigation is the most efficient irrigation. All the water comes from underneath. There's no standing or flowing water on top. All of these factors help reduce Phytophthora. I think overall growers in the Southwest over the last 20 years have greatly increased their use of drip irrigation, and we still have some flood or furrow irrigation, but the number of people doing that, I think, continues to decline over time. The only problem that we run into with drip irrigation is we can't control the monsoons and thunderstorms. So the drip, I think, is our best chance to control moisture, with the caveat that Mother Nature may or may not care how good a job we're doing. Vince is smiling. Good questions? All right. If there aren't any other questions here, I'll continue and show you some of the work that we're doing, trying to sort of rehab the microbiome so that we can go from this out of balance situation that encourages to disease to seeing if we can sort of restore some of the beneficials. And the work I'm going to show you today is mostly lab growth chamber work. We have lots of racks with LED lights and growth rooms. We probably run 15 to 20 of these racks at any given time. And the experiments themselves are really pretty simple. We grow chilies in pots, we inoculate them with Phytophthora. The disease is really, really rapid. If we keep the soil moist to encourage Phytophthora, we can have healthy chili plants looking dead like this. And these plants, this is only 7 days after inoculation in our lab. So some of the experiments we do, we take biocontrol agents that are already on the market. This one, the trade name is double nickel, and it's a single strain of a bacteria called Bacillus, and I forget which species of Bacillus it is. But in the figure here, the 4 plants on the left were just grown in standard potting soil, inoculated with Phytophthora. The 3 on the right were grown in the same potting mix, and then they were treated with double nickel or inoculated with this strain of Bacillus about a week before we did the inoculation with Phytophthora. And you can see 2 of the 3 plants treated with double nickel are doing very, very well, whereas 100% of the plants untreated are completely wiped out. This particular Bacillus was discovered probably, well, at least 20 years ago. We know it antagonizes Phytophthora. If we do some Petri dish assays in the lab, this particular Bacillus actually makes an antibiotic that kills the Phytophthora. It also occupies some of the niches that Phytophthora likes to colonize. So it's probably interfering with Phytophthora on a couple of different levels. And in this case, you can see that we keep, in this particular experiment, 2 of the 3 plants very, very happy. Now, this is going to be kind of a recurring theme. The biocontrol agents rarely hit 100%, but in this case, the double nickel in this very small sample size in this single photo, we went from 100% loss to only 30% loss. And I'm not going to pretend that those numbers translate to field, but clearly we get some protection, but it's rarely 100%. The next experiment in this slide, this is a different biocontrol agent that's actually on the market. The trade name for this one is Lifeguard. This agent is a different strain of Bacillus. It's a Bacillus just like double nickel, but it's a different strain. This Bacillus is really interesting because it works through a different mechanism than the double nickel works by. The Bacillus strain that's in the Lifeguard doesn't actually antagonize Phytophthora directly very well. But the strain of bacillus that's in Lifeguard actually interacts with the chili plant and it turns on some disease resistance genes in the plant. So plants have sort of an innate immune system, sort of like people do. I think we're all familiar when we get an infection, we may not be immune to the infection, but our bodies have a couple of different responses. And one of them is we get a fever. The reason we get a fever in people is we try to raise our body temperature higher than the pathogen is good at growing at. So humans getting a fever is a sort of an innate defense response against bacterial infection. Now, plants do kind of the same thing, except they don't get a fever, they don't have antibodies. The mechanisms are a little different, but the general idea is the same. So plants actually have two different innate response systems. One's called SAR, the other's called ISR, for systemic acquired resistance and induced systemic resistance. But what happens is that a bunch of genes that help with defending against pathogens get turned on when these systems are activated. And the bacillus strain that's in Lifeguard actually activates, I think it's the ISR system in plants without causing any disease. So in this case, you can see that even though Lifeguard doesn't directly antagonize Phytophthora, turning on these plant defense genes in the chili peppers does a good job suppressing disease. And in this particular photo, it looks like all three of the plants that we treated with Lifeguard are pretty well protected. If you look really closely with a keen eye, you'll see the plant in front, the leaves are starting to wilt a bit. So in this case, we still didn't get 100% infection. But if you're thinking about the end of my talk here, we've got one bacillus that directly antagonizes Phytophthora. Here we've got a different bacillus that turns on plant defense responses that help suppress Phytophthora. So we've already got two different modes of action that we might think about combining together to see if they would cooperate and suppress Phytophthora even further. Let's see, and I guess that's the end of these symptom slides. So we've actually tested probably close to 20 of these different agents. The two that I've shown you here are bacillus. We've got some beneficial fungi, especially out of the trichoderma family. There are other genera of bacteria, especially some from the Pseudomonad family. So a lot of what we do is go through and test a lot of these agents individually, one at a time, just like I've shown you in these photographs. And then, let's see. Well, I guess I do have some more of the symptom photos. This is 3 sets of plants. On the far right side, I need my glasses to help. We're looking at healthy plants. The ones in the middle are untreated. And then in panel E, we've got a set of plants treated with yet another strain of bacillus called Thea. And then the other 2 panels, I'm actually going to come back to those in just a second. I want to spend a minute talking about how we measure disease in the lab. And to do this, I want to walk you guys through some graphs to show you exactly what we're measuring in the lab. So if we look at this graph, starting on the far right, we have a green line that starts at 0, and about 7 days, that green line climbs up and it approaches 100%. So this green line is measuring healthy plants. And there are none at time zero because all the healthy plants are seeds under the soil. About 7 days later, we start getting seeds germinating. And by about 14 days, usually about 90% of our seeds are germinated. So we'll call this 90% healthy plants. And then we've got the red asterisk at the top of the graph, which is where we inoculate with Phytophthora. And then our green line continues over in the case where we don't inoculate with Phytophthora, all the plants would stay healthy. But when we inoculate with Phytophthora, we have the red line coming down, which over time, Phytophthora makes the plant sick and we end up getting disease loss. So this isn't an assay where we artificially inoculate plants with Phytophthora where we see that asterisk. We also do a slightly different assay in the lab where we actually plant the seeds into soil that has Phytophthora already present in the soil. This would be something like we would bring soil home from a field that actually had Phytophthora in it, and we would plant our seeds directly into this soil. The reason that we use this assay is that this is a much better mimic of what growers are going to face, because growers do not come out in July and artificially inoculate their field. They actually plant into a field in March or April that already has Phytophthora in it. So as often as we can, we actually use this kind of an assay. And if you look at the green line, this is germination and survival of plants in soil that doesn't have any Phytophthora in it. And then if we look at the red line, this is growth, germination, growth, and survival of seed that we plant into soil that already has Phytophthora present in it. And the first thing that we notice is our red line, the plants germinate, go up, and establish, but we get a lower number of established plants than we do in the disease-free or the Phytophthora-free soil. We actually never see these plants germinate. And what we're measuring here is pre-emergent death. And these are seeds that Phytophthora is actually killing as they're trying to germinate and emerge. Now, the interesting thing is the ones that don't get taken out by the pre-emergent death, they actually survive for a few weeks and then they start to decline. So this is what the farmers see in the field is disease loss on plants that got out of the ground and started growing. So this assay, when we do it in the lab, this is actually a really good mimic for what we see in the field, where if you plant into a Phytophthora infested field, you're going to get a little bit of pre-emergent death. The plants that come out will do pretty well for a while. And then after the Phytophthora ramps up, then you'll get your disease loss later in the season. So we have done several experiments trying to address a couple of the features in this graph. And I'm trying to see if I can find, there's one particular thing I want to talk about. And here, this is it. So one of the things I want to talk about is different from a biocontrol agent. It's not one of the bacillus. It's not one of the beneficial fungi like trichoderma or something like that. One of the things that I'm going to come back to the graph here. One of the things that we're looking at is to see if we can reduce the amount of pre-emergent death. And the best way that we can do that is see if we can come up with a treatment that reduces the amount of Phytophthora that's even present in the soil before we plant the seeds. So there's actually a chemical treatment that we can use to do this, or one that we think we can use. And it's actually a chemical that's approved for use in organic agriculture. So this chemical is a mixture of vinegar and hydrogen peroxide. There's a little bit of chemistry that happens and it gets a funky name. It's called peroxyacetic acid. The peroxyacetic acid is a chemical that forms when vinegar reacts with hydrogen peroxide. But again, we take a mixture of vinegar and hydrogen peroxide. And when we do this, there are several formulations of this that are on the market and they're approved for use in organic agriculture. They actually have OMRI labels. And so what we've been doing is we've been trying to treat soil that already has Phytophthora in it. And what we'd like to do is reduce the amount of pathogen that's in the soil simply by exposing it to this peroxyacetic acid. When we do that, we should reduce the pre-emergent death and cause the disease loss to happen later and at reduced levels. So some of the background work that we did with this peroxyacetic acid is we would take cultures of either bacteria or fungus and treat them with different levels of peroxyacetic acid and ask if the peroxyacetic acid would kill either the bacteria or fungi that we're testing at that point. So I've got a fairly complex photograph up here, and I'm going to see, does the mouse track over? All right, that's perfect. So what I want to look at first is this panel of E. coli. And we've got a petri dish here, and when we treat E. coli with just water or zero peroxyacetic acid, we end up getting a lot of E. coli growing on the plate. But if we only treat it with 3.9 parts per million of peroxyacetic acid, all of a sudden we've killed all the E. coli in that culture. So it turns out E. coli is super sensitive to peroxyacetic acid. So we took this assay and we did it with a couple of other kinds of bacillus, and we can see that bacillus, this particular bacillus keeps surviving. 4 parts per million PAA completely wiped out E. coli. This bacillus persists until about 30 parts per million PAA. The other bacillus on the bottom persists to about 60 parts per million. And unfortunately, Phytophthora hangs on even longer. We have to ramp the PAA up to about 250 parts per million to get rid of the Phytophthora. And even a little scarier is another fungal pathogen called Fusarium. It keeps going at 250 PAA. We have to get over 500 to kill Fusarium. So all of this is a little esoteric, and I don't want to put people to sleep, but we'll look and see what PAA does to chili plants. And chili plants actually survive PAA 250 and even out to 500. And everything here is kind of summarized in this table, where if you're green in the table, you survive. If you're red in the table, you end up getting killed. And the message, the take-home message from this table is that levels of peroxyacetic acid that don't affect chili plants end up getting rid of Phytophthora in the soil. So we've started adding this pre-treatment of peroxyacetic acid and looking to see if we can use that to decrease the amount of pathogen in the soil and increase the surviving plants. So this graph, this is data from an experiment where we tested this. And the treatment inside the red box right here, there's a green bar which is percent germination, and a red bar that's percent death. So in this particular treatment, this was the control, and this didn't have any treatment. So what we see is we got good germination, good plant establishment, which is the green bar. But in the red bar, almost everything died. And then we look at some of the treatments that we did. This is a beneficial trichoderma soil guard, plus a beneficial bacillus called Thea. We got great germination, and then we also had a very low death rate. So this is a treatment that does very, very well. And if this treatment on the end where we do oxidate as peroxyacetic acid plus a beneficial bacillus, this is another treatment where we put chili seeds into Phytophthora infected soil. We get very good germination and very low plant death. So these are some of these combination treatments where we have several different agents working together. We use a little peroxyacetic acid to reduce pathogen load in the soil. Then we add a beneficial trichoderma, a beneficial bacillus, and we get very good germination and very low plant death. So the graphs are really, really hard to look at, but here's some plant pictures from some of these experiments. The untreated control, we get a few plants hanging on. Most of the plants are wiped out. In a treatment with 2 different beneficial bacillus, we get most of the plants doing very well hanging on, and so on and so forth. So I could fill, like a good professor, I could fill the next 20 minutes looking at more bar graphs. But I think that probably puts a lot of people to sleep, including risking putting myself to sleep. So maybe this is a good time to take a little break and see if there's some questions.
You got some
All right.
from Juan. Is there a relationship with soil oxidation and fungi?
Yes. So the peroxyacetic acid that we put in, it's not actually oxidation, but it's sort of almost literally acid treatment. This has both general and specific effects on the entire soil microbiome. So I'm going to come back to this picture that had way too many petri dishes in it. So peroxyacetic acid, going the wrong direction. I would love it if peroxyacetic acid was specific for Phytophthora without affecting anything else in the microbiome. Unfortunately, it's not. When we add peroxyacetic acid to the soil, we generally knock everything down. And then some of the finer points I want to make are some things like E. coli are super, super sensitive, and other things like bacillus are less sensitive. So we don't knock everything down universally. We knock some things down farther, some things less. And earlier I mentioned the microbiome studies or the metagenome studies where we use a DNA sequencer to watch every single microbe in the soil. So we're actually in the middle of experiments now, seeing what proportion of the microbiome gets knocked down really far, which portions of the microbiome don't get knocked down quite as far, and try to characterize what happens with that. We're not all the way through the microbiome studies, but we are far enough that I can actually tell you about some conclusions that on the surface seem a little bit surprising, but when we work through some of the logic, they actually kind of make sense. If we treat soil with peroxyacetic acid, we knock microbes down pretty quickly, but the peroxyacetic acid has a very, very short half-life in the soil. It only lasts for an hour or 2 in the soil. the microbiome in the soil, when we track it, it actually starts to regrow within 24 hours of treatment. And by 48 to 72 hours after treatment, the microbiome has regrown to the same levels it was at before we treated the soil. Now, when we look at the composition of the microbiome that regrows, what we find is that the pathogens are actually reduced in that regrown microbiome. So this sounds kind of like the magic silver bullet we want to find, and it almost sounds too good to be true. But when we take a minute to think about what regrows and why it regrows, pathogens like Phytophthora make their living eating plants. And when we take a field that's that's ready to plant, there are no plants growing in it. There's no chili in it. So when we use PAA to knock everything down, when things get the opportunity to regrow, Phytophthora has no plants to regrow on. So things like Phytophthora that need a chili plant there to regrow, they end up rebounding really slowly. Other things that don't rely on plants to grow, things like the soil, the carbon and nitrogen cycling bacteria in the soil, they don't need a plant to survive on. Those regrow really quickly. So in general terms, this peroxyacetic acid treatment really does do a good job of pushing the pathogens down, and many of the beneficials actually rebound very quickly on their own. So this treatment is actually kind of doing sort of what we expect it to do. So great question. The PAA is not specific to Phytophthora. It does generally knock all the living things in the soil down, but it goes away, the PAA goes away very quickly. And after it goes away, a lot of things rebound and sort of create a new microbiome in the soil. And for everything that we can tell today, the microbiome that recovers in the soil after the PAA treatment is more beneficial and a healthier microbiome than the one that was there before the PAA treatment. And in particular, the pathogens take much longer to rebound than the beneficials. So thank you. That's an awesome question.
I have a quick question.
Yeah.
So growers use a lot of like anhydric or peak acid, and also hydrogen peroxide to clean out lines, but also to provide fertilizer. I mean, have you ever looked at any of these? Because they're actively putting them on the field all the time.
Yeah, so there are a couple of reasons to add acid to the field. Sometimes fields are just too basic and people want to optimize the pH, hit the optimal pH for plants. Some of these things, in fact, the peroxyacetic acid that's approved for use in organic agriculture, there are a couple of trade names that are available on the market now. One is either oxidate or sanidate. Another company makes one called Jet Ag. And these things, one of the original uses that they were put out for was cleaning drip lines. So sometimes drip lines get colonized by algae or other things, and this could plug the emitters. So one of the uses that this peroxyacetic acid has been used for over the last decade or so is actually just cleaning drip lines. So this is, you're exactly right. We're sort of repurposing something that was already being used, and whether it was to adjust the pH in the field or clean out drip lines or things like that. Some of these things have already been in use for quite a while. Yeah.
So I got a couple of questions that all go together. So if you were to put peroxyacetic acid into a field, how deep would you have to go? How would you apply it? How costly would it be?
Yeah, so those are all great questions, and we are in the middle of figuring those out. I can give you some sort of boundary or ballpark estimates now. With respect to cost, everything that we do, we keep cost per acre on our mind first and foremost. If I told you I can get rid of 100% of every disease, you're gonna say, wow, that's fantastic. I'm gonna get 20% more chili. And I say the catch is it only costs you $38,000 per acre. Not ever gonna matter because nobody's ever gonna make $42,000 an acre harvesting chili. So our goal, we give ourselves a little bit of wiggle room. We want to initially make sure that all of these things will be $1,000 per acre or less. And I'm not dreaming that 1,000 per acre is something that growers would be excited to do. But this is sort of our maximum, get our foot in the door kind of a deal. Our goal is to get these things down under 500 per acre. All of the treatments, we design them and we make sure that when we get them to a field, will be 1,000 per acre max. And that's just to get our foot in the door and make sure it works. And then hopefully we can optimize things and get underneath that 500 an acre. Some of the things that we're running right now are only about 300 per acre. And then the next part of your question is how to get them into the field. So in the lab, we simply top drench. We take a beaker and pour some PAA solution on. That's not a practical method to put them in the field. So we are running field tests with these now, and the method of delivery that we're using is we're running them through the drip irrigation. And we're trying to optimize a couple of different things. The first thing we're trying to do is, since these things aren't free, we're trying to get the most bang for the buck. And one of the challenges that we have with the peroxyacetic acid is that it's so reactive, it doesn't live very long in the soil. So where we're at right now is delivering PAA at about 250 parts per million through the drip. And we're actually running the drip with water only to saturate the soil first. And then we run the PAA at 250 parts per million for about 2 hours. And our hope is that it will diffuse through the wet soil pretty quickly. The little bit that we do know is that this does not adversely affect chili germination. And we've actually tried it on a couple of different crops and a couple of different levels. We can run the PAA through without hurting germination of either chilies or onions. And we can actually run the PAA that way as high as 750 or 1,000 ppm. And when we do these, the cost of the PAA treatment is, I can't remember off the top of my head, I think it's under $200 an acre. And we have some field trials going in this summer where we've got that level of PAA delivered by that method through the drip. And we're going to combine it with the beneficial trichoderma and a beneficial bacillus. And right now, the estimated cost for that combination treatment is right around the 500 an acre mark.
So does this only work as a pre-treatment
Yes.
period? It won't affect if the plant's already like in an initial stage of getting infected?
That is an excellent question, and that's a point that I probably should have highlighted before. All of these treatments are 100% prophylactic. So unlike a fungicide is a chemical that's toxic to the fungus. If you get a fungicide that can diffuse into the plant, you can actually treat a plant after it's been infected. Unfortunately, the biocontrols don't work that way. These things need to be 100% pre-infection, which when you think about the cost of treatment, requires that you calculate things differently, because a fungicide that might reverse an infection if you caught it really early, you might be willing to go twice as expensive. because you can wait to see if you need it. If you don't spend any money. Whereas the biocontrols, you don't have the luxury of waiting and deciding. These things have to be out before infection starts. Otherwise, the biocontrols just simply won't reverse an infection. So in that sense, they are a little bit different than the fungicides. Now, there may be a little bit of wiggle room in here for some very specific situations. And Phytophthora is one of those situations. So we talked about how Phytophthora gets spread through the water. And a monsoon can turn one infected plant into half of a field lost. So for the PAA, peroxyacetic acid, the swimming spores of Phytophthora are very, very sensitive to PAA. They get killed with pretty low amounts. So in the case where you've got very low level Phytophthora and you get hit with 3 thunderstorms the same week, we haven't tried it yet, but we plan to run a trial. If we get that situation, we're going to try applying PAA to the field with the goal of killing all those zoospores swimming around and getting carried around with the water. So this is one potential case where we might be able to use this in kind of a targeted fashion. But the general rule of thumb, and certainly for the bacillus and the trichodermas, those actually have to get on whether you know disease is coming or not. In a case where you didn't have any disease, you are going to spend $500 an acre you didn't need to. When disease shows up, you won't have avoided not spending $500 an acre and watch your crop melt. But the calculation for determining whether you want to spend money on a control is going to be a little different because it's not like a fungicide where you can wait and decide. It's one of these things that you have to commit to early. And the hope is that you'll avoid some disease. The hope is not that you've spent money treating a field that would never get sick. So for the particular growers, the calculation might get a little more complex if you have a field that you're coming back to after 5 years of rotation and you know that it got hit hard 5 years ago. You might decide it's worth investing in these. Whereas another field, you're coming back after 5 years of rotation and it had no disease last time. You might decide to skip these. So I think there's a lot of room for nuancing this a lot, and I'm sure growers will want to do that.
Would there be any benefit to applying these bacillus and biologicals just before for the monsoon season?
Yeah, so this is actually, it's going to be sort of disease dependent. For Phytophthora, giving the field a little bit of boost before rain, maybe throwing some PAA on after a rain. Those are things that would make sense for Phytophthora. So I have Fusarium up here on this Petri dish assay. And we've been doing a lot of work with Fusarium as well. And what we figured out is that Fusarium symptoms show up pretty late in the year. But Fusarium infection actually happens about as quick as the seeds germinate. So in this, for Phytophthora, you might be able to apply the biocontrols late, but for Fusarium, we have to have the biocontrols right there at germination. And Verticillium is probably second behind Phytophthora in damage to chilies. And Verticillium actually behaves a lot like Fusarium. And that the symptoms Verticillium symptoms don't show up until very late in the year. But the Verticillium infection probably happens almost right at germination. So overall, I think it's a good idea to apply these things immediately right off the bat. The neat thing about a biological is it's alive and it grows. So if you get the seed, the germinating seed colonized, these things should be able to grow with it throughout the year. Some cases like Phytophthora, you might be able to apply later in the year, but most diseases, you really want to interrupt them right at the start when the plant is germinating. The other thing that I'll add quickly is something that surprised us a little bit. We have done some of these assays in fields that we didn't see disease pressure in. And we actually got our treated plants performing about, I mean, it was not double, but maybe 5 to 10% better than the untreated plants. And what we think is going on there, these things are not fertilizers, they're not growth hormones. But what we think is going on there is there are other diseases that we don't normally really care about. We don't, maybe even we don't know what they are, but we think of them as athlete's foot diseases. They just sort of niggle at the roots, and in a good year they happen and we don't ever really notice it. But we put the biocontrols in, and even in the absence of Phytophthora, we get this little tiny boost. And I think what we're doing is we're suppressing these sort of athlete's foot diseases.
Just a couple of minutes.
Oh, we are,
Yeah.
wow, time flies.
So we had so much time with all this interesting discussion and great questions, so the time did fly. So maybe time for one more question for Steve.
Yeah.
Yeah,
I'd like
okay, Jeff?
to do it quickly. So I work in greenhouse situation. So how would like PAA work in a recycled water situation? Would it kill a lot of these like Phytophthora within the water before it even gets to the plants?
We have not tried exactly that, but we want to. And on paper, I always get surprised. I always say we have to test it because everything that looks good on paper, you want to make sure it works in reality. But I think greenhouse systems, recycled water, I can't think of a better situation for PAA.
Thank
You know, with that, let's thank Steve for an excellent Chile Chat.
Well, thank you, everybody. It's a pleasure to be here. And I'm sorry I was so long-winded with the answers. I didn't realize the hour and a half went so quickly.
That's what Chile Chats are all about. It's a discussion. So thank you for the information.